Hello everyone, let's discuss test 2 of zoology. So question number 1 asks us about the AFB full form. It's European Federation of Biotechnology, which is A. Next is directly from NCERT, how many restriction enzymes are isolated till now? 900 as per NCERT. Third is asking us about the nomenclature. So the first word or the first letter in the word is derived from genus, second and third from the species, fourth from the strain of the bacteria. So A is the answer. Fourth one, it's asking us about the two enzymes. So one is methylase, the other one is restriction endonuclease. Methylase does the methylation or adds on the methyl group to the whole DNA sequence, whereas RE cuts it. So A is the correct answer. Question number 5, it's saying that if a transformed cell, which is E. coli having resistance to ampicillin, is put on the agar plate. So which of the following would likely happen? So non-transformants will grow, transformants will grow, there will be no colonies, or transformants and non-transformants will grow. Transformants will grow. Fifth B. Sixth, it's asking to match it. So A is the correct answer as a fuel match. It's easy itself. Seventh is also easy. It's asking that which enzyme can digest what. So cellulase digests the cellulose in plant cell wall, protease the proteins, and chitinase the chitin in fungal cell wall. Eighth is asking that which among the following are the components of bioreactors. Actually, all of them are. So D is the correct answer. Ninth is it's asking about the functions of bioreactors, whether the given functions are correct or not. Actually, both of them are correct. And aseptic means sterilization. So both are correct. 10th is asking whether in Eco R1, Roman numeral indicates the name of the strain. No, it doesn't. It indicates the order of extraction from the bacteria. Statement 2 is correct because hint 2 does cut at a specific site which has 6 base pairs. So C is the correct answer. 11th, it's asking whether phosphate can increase the efficiency. No, calcium can. So it becomes wrong. And statement 2, retrovirus can be used to deliver desirable gene into animal cells. Yes, it can. So C is the correct answer. 12th, it's asking about the colonies and, drawing out the conclusion from whatever statements are given with regards to X-gal and beta-galactosidase. So beta-galactosidase is an enzyme which is produced by lac-Z gene, and beta-galactosidase turns the color of X-gal into blue. So when we read all of these, we get to know that colony B has recombination bacteria or the recombinant bacteria, which shows insertional inactivation, and that's the reason why it's not showing blue color. 13th, DNA is a hydrophilic molecule which cannot pass through cell membrane, yes. And the bacteria should be made competent, of course. So it's made competent by using calcium ions. So C is the correct answer. 14th, it is with regards to gene gun or biolistic gun. So first is correct, second is also correct, and so is third. So all of the three are correct. 15th, in microinjection method, recombinant DNA is directly injected into the nucleus of animal cell. Yes, it is directly injected. Microinjection method is used in oocytes. Yes, it is. And electroporation is the formation of temporary pores in the plasma membrane of the host by biolistic. No. So first and second are correct. 16th, restriction enzyme belong to the larger class of enzyme called nucleases. Yes. And bacteriophages have the ability to replicate within bacterial cells independently of the control of chromosomes also. Yes. So A is the correct answer. Next, again, easy one. We have to just match which among of these is functioning in a way. So PCR is responsible for amplification. Denaturation of DNA is related to separation of the strands. Annealing is involved with the primer, and extension is involved with polymerization. Yes, so D is the correct answer. 18th, again, if we match the plasmids, bacteriophages, cosmids, and agarose, B will be the answer. Okay, because plasmids are circular and extra chromosomal. Bacteriophages are viruses that infect bacteria. Cosmids are hybrid vector derived from plasmids, and agarose is natural polymer from the seawater. Now we have to identify what diagram this is. So this is clearly a bioreactor, and out of all the options, only bioreactor C is there. So this could have been just an easy answer. Question number 20, it's a diagram from NCERT wherein you can see that it's GAATTC, and this one is the site of EcoR1. And so here the enzyme which is used is EcoR1, and here the enzyme which is used is to attach the sticky end together, so that can be DNA ligase. So A is the correct answer. Next one is with regards to the diagram given over here of PBr322. ORI is original restriction enzyme? No, it's origin of replication. ROP is reduced osmotic pressure? No, it's replication of plasmid. HIN3 and EcoR1 are selectable markers? No, they are restriction endonucleases or enzymes. And D is the correct answer over here, that ampicillin and tetracycline resistant genes are antibiotic resistant genes. 22, recombinant DNA is a plasmid DNA that has incorporated. So 3, gel electrophoresis is the process by which DNA fragments are separated, correct? Ethidium bromide is used for staining in the process of electrophoresis, and agarose is obtained from the seaweed. Now, the answer for 22nd is A. 23rd, a limitation associated with traditional hybridization procedure used in plant and animal breeding overcome by genetic engineering is. So it's traditional hybridization that may lead to inclusion and multiplication of non-desirable genes. That's what we have eradicated from the process. 24, the scientists who cloned DNA from the Salmonella typhimurium, streptomycin resistance plasmid into the Escherichia coli plasmid. So they were actually Cohen and Boyle, and they were the foundations of the genetic engineering. 25, which among the following is a biotechnological process? Actually, all of them. Which of the following is the most accepted definitions according to NCERT? D is the absolutely correct definition of it. 27, it's simple stirred tank, which is given in your NCERT again. First recombination again was done by Cohen and Boyer in 1972. Next, we have to tell how many of these statements are correct. So asexual reproduction preserves the genetic information while sexual permits variations, correct. Traditional hybridization often leads to the inclusion and multiplication of undesirable genes. We just read that statement, also correct. Recombinant DNA technology allows us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes, also correct. Actually, all of them are correct. So D. The definition of biotechnology given by AFB encompasses traditional view, modern view, DNA fingerprinting, actually both of these. So D is the correct answer. 31, genetic engineering technique includes altering genetic material. 32, term restriction in restriction endonuclease enzyme refers to what? It refers to cutting each of the 2 strands of DNA at specific points in sugar phosphate backbone. 33, restriction endonuclease are enzyme which they make cuts at specific positions within the DNA molecule. So B. 34, it's asking about a palindromic sequence. So GATTC is the most common sequence that we've studied a lot in the NCERT, and we can directly find it out. And also it's palindromic. If you see, it's the same when read from both the ends. 5' to 3'. Whenever it's read, or 5' to 3', it's always read in the same manner. 35. We have to tell that the cutting of DNA at specific location is by the discovery of restriction enzymes. First restriction endonuclease was HIND2. Restriction enzymes belong to a larger class of nucleases. Again, direct question. 38. Dash types of enzymes are used in recombinant DNA technology to split a specific sugar phosphate bond. So sugar phosphate bond is found in DNA, and yes, restriction enzyme is the answer. Restriction endonuclease enzymes are used to cut what? So it is used to cut double-stranded DNA. Okay. Next, special sequence in the DNA recognized by RE is called, it's called restriction site, palindromic sequence, recognition sequence, or site. So D, correct, all of them. A single strand of nucleic acid tagged with a radioactive molecule, it's actually probe. 42, number of strains from which restriction enzymes have been isolated. They are 230 according to NCERT. 43, Restriction endonucleases are the enzymes that cleave the 5' or the 5' terminal nucleotides from the duplex of DNA molecules. No, it doesn't do anything like that specifically. Q says that makes sequence specific cuts in both the strands. Yes. Promote circularization of duplex? No, it doesn't. It doesn't promote that. And it says that generate 3' hydroxyl and 5 phosphate end. Yes, it does. So Q and S are correct. Third. 44th, restriction enzymes cut DNA at specific sequence. Some restriction enzymes cut DNA and to leave the same sequence. Um, some restriction enzymes cut the two DNA strands at near the recognition site. And recognition enzymes are exonuclease rather than endonuclease. Oh, we have to tell the false statement. So yes, D is the false statement because restriction enzymes are both exonuclease and endonuclease, not exonuclease, not just exonuclease. I mean, 45th, restriction endonucleases are what? So when we read all the options used for in vitro DNA synthesis, synthesized by bacteria as part of the defense mechanism, present in mammalian cell, definitely not, and used in genetic engineering for ligation, wrong. So B is the correct answer. 46. In the screening process during recombinant DNA technology, clones that metabolize B-gal turn what? They turn blue because the original color of gal is white. So then they turn blue. 47 assertion says that in gel electrophoresis, DNA fragments move towards the anode, correct? DNA is positively charged molecule, wrong. So C is the correct answer. Taq polymerase used in PCR is because it's a heat stable enzyme, and it is heat stable because it is derived from the bacteria Thermus aquaticus, which lives in the hot springs. Also correct, and it explains why it is heat stable enzyme. So A is the correct answer. 49 assertion in the biolistic method, microparticles of gold and tungsten coated with DNA are bombarded at target cells or tissues using a gene gun, correct? And is it suitable for animal cells? No, it is suitable for plant cells. So the correct answer is C again. 50. DNA fragments generated by the restriction endonuclease in a chemical reaction can be separated using electrophoresis, correct? Agarose gel electrophoresis is done. Okay, in DNA gel, the fragment of 2 kb and 3 kb will be 2 kilobase closer to the opposite end of gel, while 3 kilobase closer to the well. Yes, C is the correct answer because the heavier one is going to be near the well, and the lighter one is going to go farther more towards the anode side. DNA fragments are negatively charged. Agarose extracted from seaweeds find its use in gel electrophoresis out of all the given options. 54, it's given a diagram. This diagram is showing spooling using a spoolie brush, and the precipitated DNA is here. So yes, C is the answer. 55, what is it showing? It's showing agarose gel electrophoresis. 56, which of the following is correct match for the enzyme used to extract DNA in the following organism? So PCR, plant cell cellulase, fungus is chitinase, bacteria is lysozyme. A similar question was done above. So yes, all of these are correct. In the isolation of DNA, removal of protein and RNA is carried out by dash and dash enzymes. So proteins are digested by proteases, and nucleic acid, which is RNA, is digested by ribonuclease. 58, DNA fragments can be separated on agrogel after staining with what? So ethidium bromide is the stain. It gives bright orange color under the UV rays. 59, in agrogel electrophoresis, DNA molecules are separated on the basis of size, charge, what? Size only, okay? 60, imagine a gel through which DNA fragments have moved in response to an applied electrical current. So which is going to be farthest, it's asking. So shortest is going to be the farthest, A. 61, which of the following statement is correct in context of observing DNA separated agarose gel electrophoresis? So ethidium bromide, which is stained and gives a color under the exposure to the UV light, not the visible one, but the UV one. 62. So the criteria of DNA fragment movement is what? So the smaller fragment, it goes farthest. 63. During isolation of DNA, addition of what precipitates and purifies the DNA? So it's chilled ethanol. 64. Which protein has been produced generating a transgenic sheet that is used for replacement therapy for individuals at risk from emphysema? Alpha antitrypsin. 65th, it's asking us about the features are required to facilitate cloning into a vector. So all of these are required according to NCRT. All of the three. Next, alternate selectable marker have the ability to differentiate recombinants from non-recombinants due to. So it's due to inactivation of recombinant DNA and inactivation of enzyme. Beta galactosidase it is, not 3-galactosidase, it's beta galactosidase. So both, oh wait, this is wrong and insertion and activation is correct. So both B and C is the answer. Because insertion and activation leads to the inactivation of beta galactosidase. 67, which of the following are considered as useful selectable markers? All of them are mentioned in NCRT. 68, it's asking what the PST1 site. It's present on ampicillin, so A is the answer. 69, if our plasmid does not have an insert, it will give colonies of blue color. Then white will get converted into blue without any interference. 70, in vector PBR322, the ROP codes for what? It codes for the protein that is involved in the replication of plasmid, because that's what its full form is. 71, how are transformants selected from non-transformants? So they are selected by the presence of alien DNA into vector that results in insertion and activation, A, that, and B, that antibiotic resistance gene can also show that. So D, both. 72, presence of more than one recognition site will lead to what? It will lead to several fragments, which will be very difficult to join back. 73, the recognition site for BAM, H1 and PBR322 is where? Tetracycline resistant gene. 74. Yeah, I think this one is a repeated question. Will be changed. Actually, I'm recording before the test has happened, so will be changed. Question number 75. Statement 1. ORI sequence is also responsible for controlling the copy number of the linked DNA. And statement 2 is ORI is a sequence from where replication starts at any piece of RNA. RNA? No. And statement 3 is in cloning vector PBR322 ORI is situated between ROP and tetracycline. No, it's between ROP and ampicillin. So only one is correct. 76. Statement 1 says that presence of more than one recognition site within the vector will generate several fragments, which will be easy, which will make the process easy. No, it will make the process very difficult and chaotic. Statement 2 says that in order to link the alien DNA, the vector needs to have few, preferably single recognition sites. So statement 1 is correct. 77, asking about the incorrect statement. So, ligation of alien DNA is carried out at a restriction site present in both antibiotic resistant gene. No, it's not possible to be on both, only one. So, D is the correct answer. 78, statement 1, the normal E. coli cells do not carry resistance. Correct. Eco R1 PVU2 HIN3 are located between ampicillin and tetracycline resistant gene. Also correct. So, both are correct. If a person wishes to make a recombinant plasmid by inserting tetracycline resistant, he should put which restriction enzymes? Actually, BAM-H1 and SAL1 both. So D. E. coli cloning vector shows restriction against all of these. So D. Because it's not specified that on which section. 81. Significance of heat shock method in bacterial transformation is to facilitate the uptake of DNA through transient pores. Okay. And that is in the bacterial cell wall and plasma membrane, nowhere else. 82, biolistic is suitable for plants. It's the best method for plants, NCRT says. 83, method in which recombinant DNA is directly injected into the nucleus of animal cells by using microneedles. It is called microinjection method. 84. Method involving entry of high velocity gold particles micro coated with DNA. It's biolistics again. Gene gun. Now, during the process of gene amplification using PCR, if very high temperature is not maintained in the beginning, then which of the following step will be affected? Denaturation will be affected. 86. The synthesis of DNA in PCR will be 5' to 3'. Which of the following is related to genetic engineering? Plasmid. Which of the following step is or are catalyzed by Taq polymerase? So extension is related to Taq polymerase. 89th, primer used for process of polymerase chain reaction. It's single-stranded DNA oligonucleoside. Oligonucleoside means small nucleoside, which has fewer base pairs. 90. So it's a line of NCRT that from a single molecule of DNA, PCR can make approximately billions of copies. And in trail of this, the next answer is again billion times. 92. Which step of PCR includes the use of primer? Annealing. In annealing, we do that. Sources of DNA for PCR used to amplify a specific fragment of DNA. Drop of blood, hair follicle, fragment of skin. Actually, all of these. They all are sources of DNA. Enzyme used in polymerase chain reaction is DNA dependent DNA polymerase. So C. Final step is extension. So A. Primers are what? They are small chemically synthesized oligonucleotides that are complementary to the region of the template DNA. So A. 97. A single PCR amplification cycle involves what? It involves all of these: denaturation, annealing, extension, all of them. 98. The Taq polymerase enzyme is obtained from Thermus aquaticus. 99. We have to arrange all of these in order. So first of all, isolation of desired DNA fragment will happen. Then ligation will be done. After that, transfer of RDN into the host will be done. Then the culturing will be done. And last thing will be the extraction of product. So C is the answer. Even in fact, if you could identify that which one can be the very first step, you could have answered it. 10th, identify a character that is not desirable in a cloning vector. In an inactive promoter, an origin of replication, select table markers, restriction evidence. A is the answer, which is. So I hope this test was good for you all. Thank you.